Hi everyone, my name is Ashley Creighton, and today I'm going to be talking to you about 3D cell culture. Okay, to start off, we're going to talk about what are 3D cell cultures. They are a cell culture method that allows cells to grow in culture three-dimensionally as compared to the traditional 2D methods on plates and slides that we're all used to. It's applicable to lots of different types of research, ranging from drug discovery and delivery methods, cancer and stem cell research, all the way to just basic cellular studies that might focus on cell differentiation, gene or protein expression, or just the general physiology of those cells. So there are two main categories of 3D cell culture methods, scaffold-based and scaffold-free. First, we're going to talk about the scaffold-based methods. The scaffolds provide support for the cells within the culture, and they can be made out of natural or synthetic materials. So the natural ones might be made out of different types of hydrogels, whereas the synthetic ones might be made out of different types of metals or ceramics or glass. They should all be biologically compatible with the cells you're trying to grow. And when you're choosing what type you want to use, you want to consider their sizes, structure, and material that they're made out of. So the scaffold-free methods work by promoting cell aggregation to create the spheroids that I mentioned earlier, using three main techniques, all of which have certain different ways to prevent the cells from attaching directly to the plates or the tubes that you're using to grow them in. This is just a general schematic of those three main ways to do it. In the forced floating technique, the cells are added to the plates or the tubes that have some sort of coating that's either non-adhesive or ultra-low attachment, and that prevents them from attaching and makes those spheroids in the bottom of those tubes. In the hanging drop method, the cells are placed on a plate in droplet form with a whole bunch of droplets on that plate, and then the plate is actually tipped upside down, and the spheroids form in the bottom of those droplets. And then the last main way is an agitation-based method, and it basically works by not preventing them to attach, and because they're constantly circulating either by a machine or turning and rotating the plate to promote that aggregation. Just like the scaffold-based methods that I talked about, there are also quite a few variations of each of these methods that can be used depending on what your goals of the study are. For imaging and analysis of your 3D cultures, there are a lot of companies and even more machines that those companies make that can help with this. These are two examples that are shown here. They can give you a lot of different information, from just the overall culture of your single cells to different calculations that you might want for your samples. You can also follow methods that we discussed in class, like staining and fluorescent labeling, to get some of these images too, and I'll kind of show you that here in a couple slides too. And just like everything else with 3D cultures, it's all going to depend on your cells that you're trying to grow, your specific method of 3D cultures that you're using, and the goals of your research. So for our controls, because there are so many different ways to do 3D cell cultures, it's really hard to have just a blanket control set that you can use for all of them. And because they're still kind of being developed, there aren't a lot of reliable in vitro controls. And it's all going to depend on what method you're going to be using, cell types you're growing, and the goals of your research. And then you can kind of reach out to other groups who are doing similar work and see what they're using for controls. For general growth of the cells in 3D culture, the best control that we have right now is in vivo, usually in an animal model that's going to be able to support the growth of your cell really well. Because with that, if we don't get growth of those cells, we know that those cells aren't viable for some reason, and we can kind of start over and see what happened to our cells to prevent them from growing, even without being manipulated at all. So advantages and disadvantages of 3D cell cultures. There are a lot of both. The big one for 3D cultures, they are actually considered to be more accurate representation of how cells will grow and interact with each other and the extracellular environment compared to 2D cultures, because like these pictures kind of show, especially the one on the left, you can see those cell-cell interactions, the interactions that they're having with that extracellular matrix, and the cells themselves. And then in the picture on the right, we can see the different kind of zones within the cell cultures. So those cells in the middle aren't going to get as much of those nutrients compared to those cells on the outer edges of the cells. So it's more realistic to what's actually happening within like our own bodies. We have those natural gradients here too. They can, because of those gradients, they can give us better knowledge of how a drug might actually work in the body versus in just those 2D monolayer plates, because they're going to have equal access to all of those drugs that we're trying to use. And that can eventually prevent us from potentially using animals for research, or like if we did our drug research on those 2D cultures and we're like, yeah, this is great, but then we get to our animal models and then all of a sudden those drugs aren't working as well as they did in study on the plates because we don't have those 3D models. So this is a really good application and a really big advantage of those 3D cell cultures. Cell growth rate can be slower or faster compared to the 2D methods. A lot of times it is going to be slower right now just because it is newer and some of the methods are being optimized. But again, that is going to depend on the types of cells you're trying to grow and what you're trying to do with them. Analysis of these cultures can be a lot more difficult compared to 2D cultures, especially if you don't have the right equipment that you're using. And then because there are so many different methods to use and different parameters that might be involved in your studies, reproducibility is really difficult with these cultures right now, but hopefully that will be something that will get better as time goes on. So the first study that I looked at was using 3D neural cell cultures using a scaffold-based system to look at the effects of silencing a mutated ubiquitin variant on p-tau deposits intracellularly. This image shows the immunohistochemistry of the 3D neural cell cultures staining the p-tau deposits. You can see really well those cells growing within the scaffold and surrounding the cells. You can actually see the scaffolding system, and those darker cells have more expression of the p-tau deposits versus in that lower panel we don't see as much of the staining, so that silencing is preventing those p-tau deposits from forming within the cells. So the goals of the second study that I looked at were to look at 3 different 3D culture methods and compare that to 2D cultures to create consistent tumor spheres and how they can be used to improve cancer research in general. So in this picture, the top left panel, number 1, is that 2D cell culture. Panel 2 is the rest of the panels are all 3D cell cultures. Panel 2 is using the low adhesion plates that I talked about. Panel 3 is using the hanging drop method, and panel 4 shows the spheroid that was created in a natural scaffold system, and you can kind of see that scaffolding system surrounding that spheroid. The last study that I looked at was looking at the presence and possible formation of lipid droplets in canine osteosarcomas. This particular image shows a comparison between 2D on the top and 3D on the bottom. So you can kind of see with the 2D, those are like the cells that we're used to seeing in class. We can see like the nucleus and the lipid droplets that are formed, so that's what's shown in the red. And then in the bottom, we see some really nice 3D images of the cells, and we can kind of see those zonal differences, like I had talked about briefly earlier. So like panel F is a really good example. So there's a lot of expression of the protein that they're looking at in that intermediate and outer zones as compared to like panel G, where it's more centered into the middle of the cells. So this is a really nice example of that zonal difference. So in summary, 3D cell cultures are becoming much more widely used because of their similarities to how the cells would normally act in vivo. And even though there are some drawbacks to them, research is always moving forward, so I believe that 3D cell culture methods are going to continue to improve and get better from what they are right now. And they may eventually even replace the use of animals for certain types of research completely, which would be super awesome. Here is a list of resources that I used. Found a lot of great information on 3D cell cultures. Thank you for listening to my presentation today.